All right, in this last video for DNA replication, we're focusing on telomeres. Now, there's a video link on our links page that you should watch first so that you understand what I'm explaining here. So if you haven't done that, if you haven't watched the telomere video, stop this and go and do that first, and then come back to this video. All right, so if you've watched the YouTube clip, then you kind of have a basic sense of what it is. But basically, you have your chromosomes, right? So if I draw your chromosome, there are pieces at the end of your chromosomes that are pieces of DNA that don't code for protein. And these are the telomeres. Now, one thing I want to point out to you, because we talked about similarities and differences between prokaryotic DNA and eukaryotic, do you think that prokaryotes have telomeres? The answer is no, they don't. Okay, so because our DNA is linear, we have these sections at the very end. Right? Now, what happens is, like it was saying in the video, if these are those RNA primers, we know that in the Okazaki fragments that the primers have to be removed, right? Take the primers out, and then DNA polymerase can add new nucleotides back in, filling in these gaps, right? So that's why in this last picture, it's a seamless piece of DNA, and ligase would come back in and stitch in the holes. But you have a seamless piece of DNA. But right here, there's nothing to be able to attach the nucleotides to, because you have to have a nucleotide here with a 3' end available to bring in new nucleotides. Over here, there's nothing. So what ends up happening is this space is unfilled. So what occurs after several replications? Well, the ends of your chromosomes get shorter every single time. A little bit shorter, a little bit shorter, a little bit shorter. So like I said, telomeres are nucleotide sequences at the ends of our DNA, but they do not contain genes. Okay? They're usually a repeat of this sequence, and they can be, repeat this sequence 100 times or 10,000 times. So they could be short, they could be really long. If you happen to have damage, like a mutation or something that happens in a telomere, it would not trigger apoptosis because it's not necessarily damaging to the code of the organism. Whereas if you mutated a gene or changed something about a gene that codes for a protein, that could oftentimes be a signal to trigger cell death. Okay, like that's pretty bad to damage your gene regions. The interesting thing is that there is an enzyme called telomerase that can replace telomeres. But most cells don't have this enzyme. What they have found, though, interestingly enough, is that cancerous cells, remember these are dividing over and over and over again, and stem cells actually do have this enzyme. And germ cells that are like the beginning cells when an embryo is formed and things like that, telomerase produces really long telomeres. The reason is because those germline cells are giving rise to the embryo, and they're dividing constantly. Okay, it says that they're dividing constantly. So if you didn't have telomerase, you'd probably run out of telomere pretty quickly. Now, why would that be a problem? Well, the telomere, again, is non-coding. But let's say, you get rid of telomere and get rid, and then starting like right here is a gene. Well, every time it divides, it gets rid of that telomere, right? Eventually, you start hitting pretty vital DNA, and then the cell would go through programmed cell death. So a cell has a lifespan. A normal human body cell can divide about 50 times before it can't divide anymore. So if you want a cell to keep dividing, you need to keep replacing that telomere. Now think about this. What should be true about the telomere length of an elderly person compared to a high school student? Well, if they're older, their cells have been dividing a lot more, so their telomeres might be shorter. High school students younger haven't had as much cell division, so their telomeres should be longer. But here is something to consider. If you need a reason to relax, stress can target your telomeres. It says research suggests that telomeres are targets of stress-induced DNA damage. In other words, they are very sensitive to external stress, more than the rest of your genome. You've ever heard that saying that if you're too stressed out, you're not going to live as long? There may actually be truth to that. So everybody just needs to relax. Take a time out, take a breath, and be happy. That concludes this series of notes. Have a good day.